Good morning, my name is Mantasha Ahmed, and today I am going to explain and let you all know about how chemical foundations of biophysics will take to place, chemistry of biology, amino acids, and how electrolytes are important for us, and what is the primary structure of macromolecules, what we say, primary structure of proteins, to be specifically. No physical method would have been able to establish a complex structure because we know from the surrounding molecules come in and flow out due to the metabolism. And we know this for this how chemistry is involved. Like the we know that how urea was the first synthesized compound, and it was done by Frederick Wöhler in 1882, and how other molecules were then synthesized in chemistry. This is how chemistry and biology are interrelated to each other. Further, when the experiments took place in that time in 80s and 90s, the inventions and the discoveries, they helped in development of biological chemistry, what we say biochemistry now, and that is related how macromolecules macromolecules that are present in our body, the bonds which are present in it, how it is and what are the structure of it. This is how it focuses on it. So we will learn about it further in this video. Very important point, that is a living system is chemically heterogeneous. Chemically heterogeneous, what that means? It is pointless, like it is pointless to describe that the molecule, when biological molecule, when it is taken out of the body, like if I have some DNA in one of the cell, and if I take it out in a cell or in a laboratory, so it is not living, it will not live. So it is a chemically heterogeneous system, how all of the molecules that are present in our body, chemical biomolecules, they work together to make the cell living, to make the whole body living and work it properly. Now we are going to learn about how living matter is characterized by unity of chemical structure. Like we know that molecules are bounded together with the bonds, uh, bonded together with the bonds, and we will see how, like, these bonds make it a complete unite structure. One like a one human body, there are infinite bonds between the tissue, between the cells, between the organelles, then the components of the organelles. If we go in microscopic structure, that we will see there are carbon, hydrogen and nitrogen, oxygen. These are the four main elements of which the whole body of the organism is made up. The biological evolution, like with the biological evolution, when there was a big bang theory, so there were first inorganic molecules were made, and that small inorganic molecules, they tied up together to form one first organic molecule, and from that organic molecules, slowly prokaryotes, then eukaryotes, and like prokaryotes, ancestor archaebacteria, these all were made, and how the evolution took place and how is that the man has evolved. So from this we know that biological process and chemical process, they have biological process is preceded by chemical evolution, chemical processes. So it is inseparable to understand biology without chemistry. So it is very important to understand these things. Life is characterized precisely and subtle individualization. The minor differences in the structure of the molecule, say the differences between methyl and ethyl group, these are the minor differences. Chemical reactions that take place in our body, these are strictly controlled, like they have a mechanism which will control. We know that there are promoters, there are suppressor for certain reaction to occur in our body. So we know that all these reaction in our body, in our cell, they are done in a continuous manner, or we can say controlled manner. And as I mentioned just few seconds ago, that the carbon, hydrogen, oxygen, nitrogen, sulfur, these are the main elements which make up the body. Like these are the elements which will be found in maximum amount. But there are some micro elements, metals we can say, that are also important for us, like sodium, calcium, and chloride, ions, zinc, magnesium. These are all important cofactors which are needed by enzyme to work properly. So all these things make up our living system working, and this is how we see how um, how biology and chemistry are interrelated. So we need it for work. If we come to like the amount, so what we see, that hydrogen has an amount of 63%, oxygen has 25%, carbon has 10%, nitrogen has 2.44%. So from this we know hydrogen and oxygen are in abundance. What we know that in our body water has the maximum volume. Okay, we know that our body is made up of 70% water, so hydrogen and oxygen, they are the most present element in our body. Now we will understand about amino acid. What are amino acid? We know that amino acid uh, is a carbon which has 4 different valencies. Okay. Now I told you that about amino acid we will discuss. So there are mainly 20 types of amino acid which build up the protein. All proteins in our body, those are built up by 20 different types of amino acid. But there are more types of amino acid which does not take place in what we say protein. So they are involved in metabolism. So we know this. What is the structure of carbon? We can say COOH and H2. Okay, this is the basic structure. This is the basic structure of amino acid. Now you see this. This has 4 valencies: H, NH2, COOH, and R. This is the variable group. This keeps on changing. This keeps on changing when it is like, it can be like H, it can be sulfur group, it can be acidic amino acid, it can be a basic amino acid, then we have amine groups. Okay, so and so much. Okay. If it is this R can be H, this can be SH2, this can be COH, and this can be CH2NH. So there can be different compounds which make up this protein molecule, and these are different kinds of amino acid. Now when the amino acid is in a reaction, or in a, you can say like in a content, liquid content, so it is in a, it has a charge of positive and minus. So this form is called zwitter ion formation. All the amino acids which are formed due to polycondensation of amino acid. Like the H2O will go, like all the proteins or the polypeptide that go on, the H2O like this, COO this, the COH of this, and H of this. These were H of another amino acid, and OH of the first amino acid. This will condense and it will make amino acid. It will add on amino acid like this. Now the amino acid are classified on the basis of their R group, variable group, if they are neutral, basic, or acidic, or what we say ionic. So neutral amino acid are glycine, valine, alanine, leucine, isoleucine, phenylalanine, and proline. These all are neutral amino acid. When it comes to tryptophan, it is a aromatic hydrocarbon. It has a cyclic compound. When it comes to serine, thurine, methionine, asparagine, glutamine, cysteine, these are another one. Acidic amino acid, aspartic acid, and glutamic acid and tyrosine. Basic amino acid are histidine, lysine, arginine. So we know about all the 20 amino acids. Now our another topic is now about the electrolytes. Living organisms contain numerous small ions, organic or inorganic, cations and anion, basic and acidic groups of amino acid, residues in protein, and of nucleotides in nucleic acid. The dissociation of electrolyte. Now when these small ions, they get dissociated into ions. So this is done in due to the presence of water in their surrounding. The theory of electrolyte is the concern of physical chemistry course. Here we shall confine ourselves to brief information needed for our subject. Like I will tell you by a reaction. Like there is a H2O and it get dissociated. Like there is a water molecule in our body and it get dissociated. So H2O3 plus. Now see, this is the reaction. H2 plus H plus. This gets dissociated. H2O3. Now there are three molecules. Three molecules of H3, H3O, H3O. Okay, we know that. Now this is called the electrolytic dissociation. The water is a very weak electrolyte. It is like it get dissociated easily, and it has a very low dissociation constant at 25 degrees Celsius in water. Since H plus molecules and OH are like the hydroxyl, H plus ions are equal to the OH ions in 1 mole per liter. So, this is the equation. Now these H plus present are equal to the OH because the water got dissociated. H2O got dissociated. So H plus OH, they have same concentration, that is at 10 to the power minus 8 moles per liter. And this is very important that all the biological electrolytes, these are a weak electrolyte. These all are weak electrolyte, and this dissociate slightly and slowly. Let us consider dissociation of an acid H plus. Like there is some acid, weak electrolyte Ha, and that it get dissociated in H and A, different molecules. Like there is HA molecule, it get dissociated in H positive plus A negative. See this HA, H plus into A minus. These it get dissociated in its subsequent ion. Now the dissociation constant, equilibrium constant for this is K. This is the rate. Now when we see for this reaction, the reaction constant, this K will be denoted by AHAK, AHA. See this will be the dissociation constant, because this depends on what? This depends on the reactant and the product molecules. It depends on both. Now when we see that H ion is a derivative, like it is important, it drives the whole reaction. So we take that the small a that you see here, this is nothing but the stoichiometry to balance the ion, the equation. This is just stoichiometry. So this is the dissociation constant. Now when we see, when we see pH of this reaction, the pH of the equation, this reaction of HA molecule will be denoted by pH equals to log half K pK, pK minus log H to the concentration of A. Okay. This will be the pH. This is the pH. pH half pK minus log H to the power A. So this is how we will take out the pH if the equation is given. The ionization constant. The ionization constant is denoted by alpha, and it is dependent on the concentration of ion that have dissociated. Like the number of ion that have dissociated, I told you it is H plus and A minus. So it will be like this. Yes, dissociation constant alpha, A concentration, and plus both of the products form. This is how we will take ionization constant and pH of the equation and the dissociation. Now from this we can denote that if the value of K, this, you see the K, this K, if it goes on increasing, the dissociation, the acid is very strong. If the value of K is bigger, the value of the strength of the acid is very strong, so it is a good acid. And if this pK value goes on decreasing, then the value is then the acid is very strong. The strength of the base declines with increase of K, that is increase with increasing pK. If the pK is more, then it is more basic. If the pK is less, then it is more acidic. This is inversely related to that. Now if we come to the electrolyte now we were discussing earlier about the amino acid, we know that, and I told you about zwitterion. So we will again make the structure of the zwitterion. See, this is zwitterion. Then there is a minus and plus charge on the amino acid. This is amino acid. This is amino acid. You can see this. This is amino acid. Now these two have dissociated. Now we have to know about the ionization constant of these. When we want to know the ionization constant of these and the pH of these, like if it is a basic amino acid or acidic amino acid, or it is a neutral one, so how we take it out? There is an equation given in chemistry which is half pKa1 plus pK2, like this. See, pH is equals to half pK1 pK2. pK1 will be for this one and pK2 will be for this. So this is how we will take out the pH, like concentration of COH and NH2 amine group and the carboxyl group. This is how we take out the pH. Now this electrochemical property of amino acid proves that they are dipolar molecules, like they have two polarities, one in carboxylic acid and one in amino group that is present in that. The heat of the reaction of ionization of organic acid. Now when organic acid present in our body, they are heated, so we will know this will be the reaction. So see, organic acid is RCOH, dissociation takes place minus and H plus. This is what heat reactions are done. Now ionization of these are low, being of the order 4 kg per mole for the dissociation of substituted ammonium ion. If we see for an ammonium ion, if there is some ammonium ion present in our body, it will be like See this. For any ammonium ion present, see this, RNH2 gets dissociated in RNH2, NH positive, NH2, NH3. This is RNH3. See, this is RNH3. This is RNH3, 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 and this is RNH2 plus N plus. Now we see this. See this. 4 kg per mole. For this reaction, the dissociation constant is 4 kg per mole, and for this reaction, it is 50 kg per mole. 50 kg per mole. For this, it is 50 kg per mole, and for this is 4 kg per mole. So we know that it has the heat of dissociation for amino group are more than for carboxyl group. This is all about electrolytes that we are going to study. Now the composition of primary structure of proteins. What we say? Proteins. The proteins are macromolecules. Macromolecules composed of one or more polypeptide chains. One or more polypeptide chains made up of amino residues. One end of the chain bears a free amino group, so called N-terminal. The terminal which have NH2. The terminal which will have NH2 ending, it is called N-terminal, and the group which has COOH terminal, it is called C-terminal of the amino acid of the protein. The characteristic molecular mass of individual polypeptide chain in protein are of the order 20 kilodaltons. Like this is the molecular mass of some protein molecules, which corresponds to 150 to 180. Like 150 to 180 amino acids when they meet, it is called, it has a molecular mass of 20,000. Molecules that contain less than 100 residues are usually called polypeptides. Okay, the molecules or proteins, polypeptides which have 100 molecules or less than that, these are called polypeptides. And the molecules, amino acid, when they are jointed in a amount of 150 to 180, these are called proteins. The peptide bond. How is peptide bond formed? I told you, when the amino acids join, H2O is taken out. Now about the peptide bond. CONH. This is the peptide bond, CONH, which joins together amino acid and protein and has a specific planar structure, has been found by X-ray structure analysis by Perling and Corey. All the 4 atoms in the peptide bond lie in one plane. The NC bond is shorter. The NC bond, you see this? This is peptide bond. This NC bond is smaller because it is unsaturated bond. These are usually smaller, and these are saturated bonds, single bonds. So these, the bond length for these is more, and the bond length for this is less. This is what I am trying to say. So we know that when the H2O molecule is taken out, the polycondensation, I told you, the polycondensation, through polycondensation amino acid joins together, and when we hydrolyze it, like when we will provide water molecule to it, the protein or the polypeptide chain will get dissociated. This is what all it means. So this is how, and we know in detail, like we already studied in physiology, what is the basic structure of amino acid and how it works, and the nucleic acid, how they work and what are their function. In biochemistry also we learned it. So that's all about today. Thank you.